I hope that every one of you is fine and will remain fine. Today I am going to talk about the other stages of the pathogenesis. We already know that pathogenesis is a process or a series of events which ultimately end at the disease. The first stage of the pathogenesis is inoculation, which we already covered. When the host and the pathogen come in contact with each other, this process is known as inoculation. They not just only come in contact with each other, but they also recognize each other. And the pathogen also tries to attach itself with the host surface. But the objective of the pathogen is not completed while just remaining at the surface. The objective of the pathogen is to get the nutrient out of the host, and the nutrients are present inside the host cell. So pathogen tries to enter inside the host cell to get the nutrients. Secondly, pathogen also fear that the wind and the water can take it away from the its potential host. So in fear and in greed, it tried to enter inside the host cell. So this process of entering inside the host cell is known as penetration. And once it is inside the host cell, it tries to establish itself. So the establishment of the pathogen inside the host cell is known as infection. Although it appear very simple process, but actually it is not a simple process. Host never offer free lunch, and it tries its level best to counter the pathogen both at the surface and even inside the host cell. Secondly, the environmental condition also play a crucial role. So for these processes, the environmental condition must be remain favorable for the pathogen to proceed. So in next couple of slides, we are going to discuss how this penetration and infection process take place in the pathogenesis. We already know that the pathogen adopt two kind of movements to reach its host. One is known as passive and other is known as active. When a pathogen tries to attack the upper parts of the plant like the leaf, bark or the flower or the fruit, it's usually done with the help of some kind of the agency, and that agency actually help the pathogen to reach to the upper portion of the plants. However, the soil-bound pathogens usually adopt the active movement. The host plant usually produces signals in the form of chemicals which are released in the rhizosphere. And if these pathogens are present there, they sense the signals and move toward the potential host. Fungi and the bacteria move with the help of flagella, while the nematode move with the help of the muscles. And finally, they reach to their potential host. Every pathogen has its own mode of entry inside the host. Fungi enters into the host through the wounds, and these wounds are usually created by agriculture tools or the animals or the insects or even by the humans. So the outer protecting cover is gone and it is easy for the fungal spore, if it is present over there, to enter inside the host. But most of the fungi also enter into the host with the help of stomata. So what will happen? These fungi moves toward the stomata and fix them over the stomata and then enter inside the host. Here is the picture. Here, this is the fungal spore. And if the environmental conditions are fine, that there is a plenty of humidity and the temperature is also optimal, this spore germinate, and the hyphae emerges from this spore is known as germ tube. So this germ tube fix right over the stomata and form a bulbous structure which is known as aprisorium. Here there is a very high turgor pressure, and this turgor pressure help the fungi to penetrate inside the host cell. And the first hyphae emerges from the aprisorium is known as infection peg or the penetration peg. So through penetration peg, the fungi enter inside the host cell. After that, a root-like structure is formed all around, which is known as hostorium, and the plural is known as hostoria. And the function of the hostoria is same like the roots. They take away the nutrient from the cells, and these nutrients finally reach to the body which is present outside the host cell. So this is how the fungi completed its process of penetration in most of the plants. Bacteria also enters into the host cell either through the wounds or through the natural openings. Most of the bacteria enter into the host cell through the wounds, and these wounds are created by agriculture tools, by the animals, or by the insects, or by the human beings. Later, these wounds are occupied by the bacteria, and these wounds help the direct entry of the bacteria inside the host cell. But bacteria also enter into the host cell with the help of the stomata. These are the natural openings present on the leaf surface. But bacteria need a film of water and the optimal temperature to enter inside the stomata and later inside the host cell. Bacteria can also enter inside the host cell through the hydathodes. Hydathodes are the openings which are present at the margin of the leaves, and then bacteria, because lot of water is already present over there, this help the bacteria to enter inside the host cell. Bacteria can also enter inside the host cell at the openings which are present at the base of flower. Through these openings, usually flower exudates its nectar, and later the bacteria also follow the same path to enter inside the host cell. Lenticels are the other openings which are present on the fruits, on the stem and the bark, and bacteria can also enter inside the host cell through the lenticels. Penetration of the nematode inside the host cell is with the help of stylet. Stylet is a syringe-like structure through which the nematode obtain the nutrients. Some of the nematode remain outside the host cell and they are known as ectoparasite, while the other nematodes remain outside as well as part of their body remain inside. They are semi-ecto or the endoparasite, and then there are the nematodes which completely enter inside the host cell. They are known as the endoparasites. So basically, penetration in case of nematode is done by the stylet, and through the stylet they absorb the nutrient present inside the host cell. Plant viruses never enter inside the host cell by the natural openings. They never enter inside the host cell by stomata, by the lenticell, or by the nectathode, or by any other natural opening. They can only enter inside the host cell through the wounds. And those viruses which enter into the plant cell through the wounds are known as mechanically transmitted viruses. But most of the viruses enter inside the host cell by the help of some kind of insect which is known as vector. So what will happen? The vector pick the virus, and some of the viruses remain at the stylet, or some of the viruses enter inside the host body. But whatever the case, later the same vector drop this virus straight into the host cell, and then the virus proceed and use the host machinery for its own multiplication. Entering inside the host cell is not a success story, but it is a part of the success story. If the environmental condition remain conducive, and if the pathogen does not produce any kind of antimicrobial compound, then the pathogen of course establishes itself inside the host cell. But it vary from pathogen to pathogen that how much time it take to establish itself in the host cell. It may be a very short duration, or it takes time to establish inside the host cell. But once it established inside the host cell, the pathogen tried to take away the nutrients. So pathogen vary when it comes to the mode of nutrition. There are the pathogens which are known as biotroph, such as the viruses, which don't kill their host and remain inside the host and take advantage of it. Then there are hemibiotrophs, which behave like biotroph means that they live in the living cell, but after that kill the living cell and start living on the dead cell. And then there are the necrotrophs, which first kill the host cell and then start living on the dead cell. So most of the fungi belong to the necrotroph group. So once the pathogen starts sharing nutrients with its host, what would happen? The physiology of the host plant is disturbed, and this disturbed physiology can be seen in the form of symptoms. We all know what is the symptoms. Symptoms are the expression of the infected tissue. In some diseases, the symptom appear after 2 to 3 days, and in some diseases, they take a week or 2 weeks to appear. So those symptoms which take even much longer time, for example, in case of few viruses, they take 2 to 3 years to appear. Such symptoms are known as latent symptoms, and the infection which produces such kind of symptoms is known as latent infection. But this is only happen in case of the viruses. But one thing you must also remember, the symptoms we usually see on the leaves, as on the right side of the tissue, a necrotic brown area. It is not appear immediately, rather it takes time. If you look at the left side, you can see that there is the yellow patches are appearing. These yellow patches later converted into these brown spots. So symptoms not appear suddenly, but they appear with the time, and they change their color as well as their shape. So this slide is just sum up what we have talked about in the last 2 3 talks. We already know that the pathogenesis is the series of events which could lead to the disease. The first stage of pathogenesis is known as inoculation, where the host and the pathogen come in contact with each other. After the contact is completed, both recognize each other and pathogen attaches itself with the host. If the conditions are suitable, the pathogen try to penetrate inside the host cell, and if the process of penetration is completed successfully, then the pathogen established inside the host. The establishment of the pathogen inside the host is known as infection. At the infection, the pathogen starts sharing nutrients with the host, and this disturbs the host physiology, which is shown in the form of symptoms. So, the time taken between the inoculation and the appearance of the symptom is known as incubation period. And the incubation period vary from disease to disease and pathogen to pathogen. This incubation period is also affected by the prevailing environmental conditions. If the environmental conditions are suitable for the pathogen, then this incubation period is short, and if the environmental conditions are not suitable for the pathogen, then this incubation period extended. So at this point, at least from inoculation to the symptom appearance, we have an idea how this pathogenesis process is gone on, and what is the incubation period, and all the relative terms, we have a proper grasp on it. So stay happy and blessed. Bye.